We are Group 11 presenting Article 1. Now, let me break it down to a few keywords. Catechol and rasosinol are both isomers of benzene. Catechol is applied in industries such as polymerization inhibition, photography development, lubricating oil, and pharmaceutical, while rasosinols are widely used in manufacturing of phenolic resins, pharmaceuticals, dyes, and pesticides. However, it is toxic and persistent of pollutant in the environment of both of these components. It is toxic to aquatic animals, even at lower concentration. Some previous studies have demonstrated using paper-based biosensor for detecting phenolic compounds such as dust, and these studies employed enzymes like tyrosinase and lactase and chromogens such as MBDH. However, the issue is that the specific response of the biosensors to pollutants such as that in real-time water samples has not been widely reported. That is why when we study further on this article, we realize that this colorimetric bioassay by horseradish peroxidase showed some limitation in terms of parameters. For instance, we need to determine the LOD, the optimum pH, the shelf life, and validation. And in terms of components, we need to consider three things: the optimum catalyst, and the optimum chromatic antigen, as well as substrate. These are few examples of good case practices in previous studies.
My name is ANISA and I will discuss the significance of this paper-based biosensor. This paper-based biosensor is produced to detect the phenolic compounds such as catechol and resorcinol in order to meet the need of a rapid and easy detection of phenolic compounds in wastewater. This biosensor uses 10 microliter of 15 microgram per mil horseradish peroxidase, which shows that only a small volume needed for this biosensor to work. Furthermore, the chitosan entrapment and glutaraldehyde cross-linking immobilization method is an efficient and easy method to be applied. In addition, by mixing components in chitosan and depositing it on paper, it is easy and time-saving. Next, the biosensor does not require pH maintenance due to the fact that the working pH of biosensor and water sample is similar. And last but not least, the application of paper as immobilization matrix allows the construction of biosensor to be cheap, portable, disposable, and stable. And thus, this produced biosensor able to meet the need mentioned before.
Colorimetric paper bioassay has numerous applications in real life due to its simplicity and cost effectiveness. Here are some of the applications which include the medical diagnosis. Colorimetric paper bioassay can be used to detect various medical conditions, for example, diabetes, HIV, and cancer. For example, a colorimetric paper bioassay can detect glucose level in diabetes patients by measuring the amount of glucose oxidase present in the sample. The figure above shows the working principle of coloriassay for the detection of glucose. Next, it can be used for the food safety. Colorimetric paper assay can be used to detect harmful contaminants in food, such as bacteria and toxin. For example, a colorimetric paper bioassay can detect the presence of E. coli in food sample by measuring the amount of beta-galactosidase enzyme present. Last but not least, it can be used for environmental monitoring. Colorimetric paper bioassay can be used to monitor water and soil quality as well as to detect the pollutant in the air. For example, a colorimetric paper bioassay can detect the presence of heavy metal in water sample by measuring the amount of peroxidase enzyme present.
Hi, my name is Kuek. Let us talk about the mechanism of paper-based biosensors. This biosensor basically made up from paper substrate, a filter paper, biorecognition elements, horseradish peroxidase, HRP, and a transducer. In our studies, HRP is immobilized through four methods, which are physical adsorption, entrapment in chitosan on paper, and covalent coupling on chitosan modified paper or an unmodified paper. The best immobilization methods is then evaluated by colorimetric assay which further analyzed the color intensity formed by phenolic compounds using ImageJ software. From the results, the HRP enzyme entrapment in chitosan on paper has been selected as it promised longer retention time and better microenvironment that maintain the enzyme catalytic activity. The mechanism will begin with the arrival of phenolic compounds, either catechol or resorcinol, a hydrogen peroxide and MBTH on the biosensor. The HRP enzyme catalyzes the oxidation of benzene diol to benzosemiquinons, which further reacts with MBTH to produce pampine products. The colorimetric image will be produced by the scanner every 15 minutes and ImageJ software analyzes the color intensity forms. The more intense color intensity determines the higher phenolic compounds concentration found from the samples. For resorcinol detection, this biosensor enables pampine to dark orange formation with limit of detection 10 mg per liter. For catechol detection, this biosensor enables pampine to red formation with limit of detection 50 mg per liter. The sensitivity of the biosensor can be improved by conducting optimization tests on concentration of enzyme, chromogens, hydrogen peroxide, and pH.
As a conclusion, we have covered from the issue to resolve, which is the specific response for biosensors to pollutants in real-time water samples, covered also the significance of biosensor addressed in the article, which is to optimize to meet the need of a marketable, rapid and easy biosensor, and the application that is used for medical diagnosis, food safety and environmental monitoring, and the working principle, whereby this technique is found to be the most stable enzyme immobilization for phenolic compound determination, since this immobilized enzyme produces a higher color intensity via the chitosan entrapment with glutarate cross-linking.
The last part of this video will be about the performance of the biosensor. My name is Lim Jing, and I'll be explaining some of the constructive ideas from our group on how the performance of this biosensor can be improved. So the first idea is regarding the colorimetric assay. We suggest to increase the drying time of the Wattman-1 filter paper from 15 minutes as stated in this research to at least 20 minutes. This can allow proper drying of the filter paper to ensure that the color intensity is stable for ImageJ software scanning. The second idea is regarding the construction of the biosensor. So in this study, the researchers used 0.01% of glutaraldehyde as a cross-linking agent for the entrapment of MBTH in chitosan. A higher concentration of glutaraldehyde can react with MBTH to produce an intense milky white coloration, but at the same time, it can also decrease the performance of the biosensor. So we suggest to use a lower concentration of glutaraldehyde, such as 0.005% as the cross-linking agent to ensure the performance of the biosensor is well maintained. The last idea is about the shelf life of the biosensor. So the researchers in this study did an analysis on the percentage decrease in colorimetric intensity of the biosensor every 2 days. We suggest to do this analysis every day instead of every 2 days to allow a better determination of the shelf life of the biosensor. Also, we suggest to keep the biosensor under cool and dry conditions to ensure that the biosensor doesn't react with other chemicals or physical agents which can contaminate or affect its efficiency. Here are the references that we use for this video assignment. And this brings us to the end of this video. Thank you for watching.